Welcome to Recovering Evangelicals, a podcast for people who were once very comfortable in the Christian faith until the 21st century intruded and made it very hard to keep on believing. I'm your host, Luke Jansen.
And I'm Scott Dyer.
I was out one day in a kind of shady part of town. I'd been told not to go there, but I somehow just took a wrong turn somewhere. I was minding my own business, but I got a bit hungry, so I started looking for a place to find some fast food. And all of a sudden, this guy jumps me. A kid, half my size, with a little pocket knife in his hand. We got into a bit of a scuffle, throwing each other around against trash cans and a telephone pole. I think we dented a couple car doors. But he eventually manages to poke that little pocket knife into my ribs. And he must have hit something important because I really started feeling woozy at that point, and completely out of breath. The kid took a few steps back from me, and so I hunched over a mailbox and I tried to catch my breath. But then I saw the kid fumble around in his pocket, and he pulled out a syringe needle. And before I knew it, before I could get ready on my feet again, the kid takes a syringe and stabs me in the neck. And that's when things turned a whole lot worse. I wasn't able to think straight. I was really unsteady on my feet, and I kept blanking out. I remember just seeing images of the neighborhood, little bursts of awareness. I was walking down the street, not knowing where I was going, and then I realized it was the kid. The kid was leading me like a dog on a leash. Before I knew it, he had led me into a basement somewhere, some kind of a building. I don't know. I couldn't make out what was going on. And I only remember finally just laying down because I was exhausted. I couldn't move. And then the kid puts this thing beside my leg, some kind of an animal. And then he leaves. And I can hear him locking the door, and I heard a chain, and then some loud noises at the door, like he was piling stuff at the door, so I couldn't get out. And then this animal at my feet starts eating my ankle. I couldn't do anything. I was barely able to even kick the animal away, and I passed out. And I'm not sure how long I'd been unconscious, but it must have been a while because I could see the animal had eaten big chunks out of my leg. It had its head inside my leg, chomping away at it, and I couldn't do anything. It was like I was paralyzed, and at some point I died. I mean, dead. And I know I was dead because somehow I saw that my leg was now completely gone. I saw the animal. It was now 10 times bigger. It had grown legs and wings, and it was now wriggling its way out of my chest, just like that alien in the movie with Sigourney Weaver.
I'm hoping that you're going to say that then you woke up or something along those lines, or I want to know where this part of town is because I want to make sure I don't hang around there.
Actually, Scott, no, this was not a dream. But then again, neither was this reality. I had wanted to tell our listeners what we're going to be doing today. As you know, we're going to be looking at the predation strategy of the Emerald Green Wasp, but all my attempts at writing an introduction just kept coming across too academic. So I thought, let's make this a little bit more personal. What if I pretend that I'm a cockroach, not just the preferred meal of the Emerald Green Wasp, but the only thing those wasps eat? And I'll describe what the predation strategy looks like from the cockroach's point of view. Sort of whet the audience's appetite for what's coming in this episode. And I think you'll have to admit, I got your attention, right?
Yeah, for sure. Yeah, it's totally.
So with that silliness now behind me, let's get back into full academic mode. Today we're going to talk about a truly grisly phenomenon of nature. Parasitic wasps, of which some say there are as many as 600,000 different species. And I'm not going to mention even some of the names, not for privacy reasons, but because they're all long and unrecognizable Latin names. And again, there's so many of them. One reason that there's so many of these species is that each one focuses on one specific prey animal, usually other insects, and even a specific prey animal in a specific life cycle stage. For example, one parasitic wasp will attack a certain prey, let's say a butterfly species, when that butterfly is still in the larva stage, while another parasitic wasp will attack in the caterpillar phase, and yet another parasitic wasp will attack it in the full butterfly stage. And then multiply that by the number of butterfly species and the number of types of insects, let alone butterflies, let alone other forms of animal species and even plant species. That's the family of parasitic wasps.
Yeah, pretty gruesome, in a sense. When you look at nature, typically we think about the beauty of nature, and I know that I do, but, there's another side to it, right? It's
Right.
Pretty ugly.
And parasitic wasps, you thought that was ugly, but they use all kinds of attack strategies to get their prey. We're going to focus on one today simply because it is so gruesome and grisly and otherworldly, just like the story I started off this episode with. This one will be the Emerald Jewel Wasp, aka, in the scientific world, Ampulex compressa, which kind of looks like an ant, but it has this shiny, bluey, greenish color, not the yellow and black stripes like other wasps, and it looks like it's made out of metal. And part of its predatory strategy is to get its egg into a living host, which becomes a food source for the larva while the larva gestates and develops, again, just like in my story, and then bursts out of what's left of the consumed shell of the host, just like that famous or infamous scene in Ridley Scott's franchise of movies, Alien in 1979, Prometheus in 2012, Alien Covenant in 2017, and Alien Romulus in 2024. Which collectively have generated over $1 billion, $1 billion in worldwide box office revenues.
Yeah, I haven't seen any of them. I'm too scared.
Oh, Scott, come on. These are classics. You can't do that.
I haven't seen any of them. I'm not even kidding. I don't like alien horror.
Scott, I'm speechless.
I know. It's like somebody saying to me that they haven't seen Lord of the Rings.
Yes. Well, so then I had a few questions for you, but now I know this is going to be this isn't going to work. Did you know there's a pseudo-scientific name for that alien in the franchise of movies?
Yeah, I did know that. Yes.
Xenomorph. Yeah. And did you know what that means?
No.
So I took Latin in high school, 2 years, grade 9 and 10, and even that much was enough to tell me that it just means strange life form. But they also, the people in Hollywood also created a more precise quasi-scientific name, Internusivus raptus. I'm going to try that again. Internusivus raptus, which I had to Google it. It turns out it's Latin for murderous thief. Now, in the movie, it's also called Xenomorph XX121, because, now you wouldn't know this, Scott, but some of the listeners might, because the fictional company that generated these things, the company called Weyland-Yutani Corps, they went through 121 iterations of these killing machines that they were trying to develop before they landed on this one that the movie now focuses on. You might remember a scene in one of the movies where the humans are walking past glass jars full of various mutations on the alien species, or I'm actually wondering if I'm confusing that with a very similar scene in District 9. Did you see that one, Scott?
No, I've seen District 9. Yes, and actually I like District 9, but no, that's not from District 9.
Well,
so.
It is. There's a scene like that in District 9, but yes.
Oh, is there? Okay.
And here's some trivia for you, Scott. Again, I wanted to see if you knew the answer to these questions, but Scott has not seen the movie. But this is then for our listeners. In one of the movies, they refer to one of the alien progeny as Cain's son. And I was wondering why. And I was going to ask Scott, but here we go. So I had it in my head, maybe it had something to do with the two brothers in Genesis chapter 4, Cain and Abel. Cain being the murderer who kills his brother. But, listeners, why is it called Cain's son? Because it's the alien that burst out of the chest of Thomas Cain, the executive officer on the USCSS Nostromo, and then went on a vicious killing spree that wiped out the whole Nostromo crew in just a few hours.
I certainly know about the bursting out thing, because I've seen that in clips many times. But
well, there you go. Okay, enough of this sci-fi trivia. We need to get back to the episode at hand. Again, today we're going to focus on the fascinating and disturbing predation strategy that evolved in the Emerald Jewel Wasp, Ampulex compressa. And to do that, we've got a true expert. Dr. Ryan Arvidsson is currently teaching and doing research in the Department of Biochemistry at Case Western Reserve University. One of his research interests has to do with certain components of the jewel wasp's venom, and another focus is on certain specific parts of the cockroach brain, which is the target of the jewel wasp's sting. And Ryan did his academic training with Dr. Michael Adams at the University of California at Riverside in the Department of Entomology, the study of insects, looking specifically at the jewel wasp and its relationship with its cockroach prey while he was there. So we could hardly do better in getting an expert guest to talk with us about this really amazing story. Ryan, welcome to our podcast.
Thank you very much.
Welcome.
So what got you into this field?
When I was an undergraduate, I was a biochemistry major, but I wanted to be a neuroscience major. So
Okay.
as I got towards the senior grade or whatever, back in the time I was getting closer, the momentum I had already maintained too many classes, and I just couldn't switch majors at the time. So I applied to UC Riverside and got into the biochemistry graduate program, but I was looking for labs that worked on neurosystems. And so I found Mike Adams' lab, and he was working on mostly molting behavior in insects and a hormone called ecdysis triggering hormone that they have a lot of good work on. But he continued to maintain interest in venoms. He had a long history of working with venoms, and he had this wasp project that a previous grad student had just graduated. And he said, well, if you're interested in neuroscience, so you got this wasp that is manipulating cockroaches by injecting venom directly into its brain. Would you be interested in that? And I said, well, yes, I would. So I ended up doing all my rotations in his lab, and eventually, kept at it. And actually, him and I are still in touch. We're collaborating on things here and there, the best of my ability. So we ended up with a pretty good working relationship. It was a really good experience.
Wow.
Okay. Something tells me you've been stung more than once.
I have never been stung.
Oh my goodness.
That is a great question. I have had my hands in with these wasps innumerable times, and never once have I been stung.
I've seen that. People moving a nest of bees or wasps, they move them, they open them up, and it kind of stir things around, and the bees aren't stinging them. The wasps aren't. Yeah. It's amazing.
Yeah, I did get to learn them for a while, and when I was in the insectary, there were other species in there, sometimes houseflies. And if they would get out, I would have a housefly buzzing around my head while I had my arms in with the wasps, and that made me real jumpy for the first year or so. Towards the end, around year 5 or 6, it was second nature.
Right.
But at first, it was weird.
Maybe you should do some research into yourself. Maybe you have some natural resistance to these.
Yeah, well, just like other parasitoid wasps, wasps have a bad reputation because of the yellow jacket and the ones that ruin people's picnics.
Yeah.
But the parasitoid wasps tend to only sting for defense, and you really have to aggravate them and hold them down. And Ampulex, strange enough, if you do hold it, its first defensive move is to make a buzz with its wings. You'll feel it. It'll make a buzz, you get a knee-jerk reaction to let it go. Yeah. I felt that a number of times.
Okay.
Okay.
All right. So let's dive into the science. I want to start off with a very high-level perspective on wasp evolution, wasps as a species in general. And I had assumed that scientists didn't get too much on that from fossil layers, fossils in the rocks, mostly because of their very small size and also because they don't have bony skeletons. But I found out that would be wrong.
Yeah, there's plenty of fossil record, yeah.
So tell us about the fossil record, about the amber, and about the genetic records.
I won't know any more about that than you do, honestly.
Okay.
But from what my work has in looking at the molecular phylogeny of a lot of these venom proteins, at least I can say from the small study that I've done on the evolution of the venom, it has a lot more in common with early wasps and ants than it does with other parasitoid wasps. One of the main model parasitoid wasps was called Nasonia, and it didn't have much in common with Nasonia. It had more in common with ants on that earlier branch. There's nice papers that show the tree and where the apocrita kind of come out. So you'd be surprised that it has less to do with other jewel wasps and ectoparasitoid wasps and more in common with ants.
Yeah, I didn't know that either.
Yeah.
But I'm sure you would have seen some images of these things in the amber, and of course, similar. Have you seen images in the fossil record?
Oh, it would be great to have as just a curiosity, just to see them. Yeah, there's absolutely. You'll see this ancient parasitoid, the ichneumid wasps with this really long ovipositor. Ancient strategies for reproduction.
Yeah, and when you say
I'm gonna go Jurassic Park here. Have they been able to extract any DNA from those amber?
Oh, not that I know of. No, unfortunately, I don't
Okay.
think such a thing's possible. But it would be neat. I would be excited to hear from it. But it's a sclerotized tissue, and that's all you're going to see. All the soft tissues.
Right. And these things go back all the way to the Jurassic period, and
before,
that's 250 million years ago, 240 million years ago. So they've been around. And do you have any reason to say that we can already see it in those specimens in the amber or in the rocks? You can see them, these bees with the stingers. That whole strategy is, for 250 million years.
Yeah, so the stinger is a modified ovipositor, and insects lay their eggs via some mechanism. An oviposition is typical. And the modified ovipositor, at least in this wasp, one of the interesting videos we captured, which I didn't know would happen, is that it actually moves its stinger up and out of the way and delivers the egg through a different out of the.
So it has two stingers?
Well, it turns out not only the one, but the stinger apparatus gets moved up and out of the way. You can see it kind of nicely move, and then the egg gets secreted right out of the oviduct directly.
Cool.
Yeah. Yeah, and then the ones that sting for defense it's much more, it's hardened and barbed and for attacking,
And so parasitic wasps not only go back so long, as I said, 250 million years, but there's also hundreds of thousands of parasitic wasp species.
Yeah, as far as I know, every insect I've ever looked up has a parasitoid wasp that hunts at spiders too.
Yeah, and they're very specific.
Wow.
They only hunt that one prey.
Yeah, and plants. There's significant parasitism of plants by these wasps as well. Some of them are very tiny, so small you wouldn't be able to see them.
Okay, so again, still before we talk specifically about the jewel wasp, let's still talk about this evolution of the wasp family in general. There's 3 or 4 main evolutionary adaptations that we'll talk about that I'd like to hear if you can say anything about. The wings, or the famous wasp waist, which seems to be a very, the fashion industry is all about the wasp waist, and the stinger itself, maybe the venom gland and the sac that goes with it. How did each of these evolve?
So how things evolved. In fact, I don't want to say that I don't believe in evolution, but using it as a metaphysics for the why of things, the answer always just comes down to selection, mutation and natural selection.
Right.
And when you multiply that by 250 million years, that's how it comes about, which is fascinating to me because the evolution of, this jewel wasp or the other wasp, their behavior is innate. They don't learn, how to parasitize their prey in particular. They have evolved that way from whatever they've evolved from towards a new target.
Yeah.
Well, that's fascinating, but I have no idea how to ask that question scientifically.
Right. Well,
philosophically, I love it. I love the idea and I'm compelled by it. I think about it all the time.
Yeah. Well, so
I'm kind of curious. And by the way, I'm a total layperson in regard to science, as much as I love science, but that's not my background. Mm-hmm. So I'm the kind of outside voice in the discussion. But one thing I have to be a bit curious about is why did it reach this point of kind of stasis where for the last how many hundreds of millions of years, I'm thinking it probably hasn't changed radically. So why does that happen? Or why, what were the thoughts on that?
Stay away from the why. I'll just say that what is fascinating to me, what I think is the case, what I would assume is that it's quite successful and you don't change something that works. And its success is predicated on the cockroach's success. And if the cockroach does well and there's plenty of them, then why change? And if it continues to succeed and goes through its life cycle, as long as cockroaches survive, this wasp can. And one thing I can say about that, though, is it's not as perfectly adapted as the cockroach because the cockroach is cosmopolitan and everywhere there are humans, there is the species. But the wasp can't live outside neotropics. It has to have the warm and the humidity. And if, for example, I'm here in Ohio, while we have really nice summers, the winters would have killed it off, but the cockroaches seem to be fine. Yeah.
Yeah. Right.
If I could give an analogy for you gave a great, more scientific answer to Scott and his question, but an analogy that I often use with that kind of a question is basically how we used to record information. We humans used to record information. We long were scribbling things in clay with little pieces of stick, but eventually we learned how to use pencil and paper, and then that evolved into pen and paper. But then we jumped into, there's that whole evolutionary adaptation where we learned how to do this digitally, and now we use things like computers and what have you. And yet we still use pencils. We still write with pencils because, as Dr. Iverson said, it works. It works well.
Yeah. Mm-hmm. Yeah. There's still value to it.
Yeah. Yeah.
Yeah.
Okay, so the wings, I was thinking they were long before the wasp family became a family. I mean, so they're related to ants, and ants also have these wings. And
that's
bees,
right.
I think bees were more recent, but that's where something before we had wasps and ants had already developed these wings. The wasp waist is important because a lot of insects can't do what the wasps do simply because they don't have the wasp waist. I don't know if you want to talk about that at all.
All I can add to it is that, where it's going to position its egg is going to depend on what it can do with its abdomen.
Yeah.
And these things aren't typically stinging for defense the way you might think wasp would. So if you watch because that's what I know is Ampulex compressa. The name compressa comes from its ability to move its abdomen, and without that free floating, well, floating's the wrong word, but without the articulation that it can do, it wouldn't be able to deposit the egg. Not in the way that it does, I suppose.
Yeah. So it's just simply it's a greater degrees of motion, and without that, they just wouldn't be able to do the singing.
That's what I'd suppose. Yeah.
So last week we talked to an expert on snake physiology, and again, there we're talking about fangs, hollow fangs that conduct venom, just like these wasps can conduct, well, eggs or venom, depending on what species we're talking about. But in the case of the snakes, they also had to adapt these sacs. They converted their salivary glands into venom sacs, and then some tubes that connected the sacs to the fangs. So here with these wasps, you've got an ovipositor. Was it first an ovipositor or then a stinger? Was it defense first or was it predation first?
I have to always qualify with these evolutionary, but with the qualifier that I'm supposing. What I know, at least from my dissections and my understanding of the anatomy, there is a direct path from the ovaries to the ovipositor area. Then in this wasp in particular, the ones I dissect, the modern wasps, if you will, they have a distinct venom gland that culminates in a sac with a ductus venatus, it's called, and that goes to the stinger and delivers venom. So there are two distinct anatomical structures, one for eggs that connect to the ovaries and one to the stinger. Where it separated into two distinct things, I don't know, but from my understanding is the eggs. What came first, the venom or the eggs? The eggs came first.
Yeah. Okay. All right. Now, one other thing that I think we need to talk about, we're still structure and anatomy, but now we're going to shift to the cockroach because we're going to use this term ganglia, and a lot of people don't know what a ganglion is. So let's talk a bit about the cockroach brain and what these ganglia are.
That's a first question when I have a new student is that they have never considered that a cockroach may have a brain. And so that's how we define it. Well, it's the nervous structure inside the head capsule. A little different than ours is the cell bodies form a sphere, if you will, where there's a blood-brain barrier that separates the brain from the hemolymph, where they have an open circulatory system and then a blood-brain barrier. And then after a few layers of that, you have the cell bodies of the neurons, and then they make connections to each other in the middle. And that's called a neuropill, or some people say pile, neuropill. And that constitutes a ganglia. And inside the cockroach head, there's the complex, the structure, what we call the brain, and it integrates the optic lobes and information from the eyes and the antennal lobes for olfaction, and then a central complex where all that information is integrated, and then sends it through various cerebra, and then it goes down these giant axons into the thorax. Their strategy for propagation of the signal is a wide diameter axon instead of insulation with myelination. And so they're really, really easy kind of to see and easier to work with. And so these signals, after a decision is made, and I use decision kind of weakly there, an integration of some signal, is the cockroach hungry or not? Is it light outside or not? Is it going to locomote or run or not? Simple decisions to make are going to be influenced by the brain and the integration of sensory data, and that's where I get particularly interested. But then it sends permissive or inhibitory signals to the thorax. The thorax has 3 ganglia in it as well, collections of neurons organized similarly, and they control the central, what are called central pattern generators. So you can get a cockroach to walk without a head, but it won't do it on its own. You have to poke it, and if you start making it go through the motions, the legs will coordinate with each other, and it will walk,
right?
But it won't be alive, as it were. It's still just, at that point, I suppose you can call it automatic or an automaton or something like that. And then the same down in the abdomen, there's several ganglia in the abdomen that control the organs and send feedback to the brain. And it's a collection of small ganglia all the way up. What we would have is a spinal cord. They have a collection of ganglia to control separate functions that is integrated with the brain and does control behavior.
Right. Many brains, many into one target.
Yep.
And one last anatomical question, if I can call it that, just to give the listeners a sense of the size here, the relative sizes of the wasp versus the cockroach. It's I liken it to a 150-pound flyweight gladiator with a stiletto fighting against a big sumo wrestler wearing a parka.
Yeah, okay, sure. Yeah, the wasp is considerably smaller, but not diminutive with respect to it. Yeah.
Right.
Yeah.
So, and the cockroach is big, and it's got this heavy shell.
Uh-huh.
It's something that's pretty hard to stick a stinger into, so it's not an easy feat.
Oh yeah, the wasp kind of takes advantage of that shell by using its mandibles to grab it, and then it takes its articulate abdomen up under and finds a vulnerable spot.
Yeah, right under the chin.
In between the sclerites, the armor. Yeah, one in the thorax and then one in right underneath where there is no armor. So the pronotum is this big armored piece of chitin, I guess, and it would not be able to sting through that, but it can hold on to it. It can grab it and hold on and then sting it from underneath.
So you suddenly got an image in my head. It's the image of Achilles and the Achilles heel. And the logic there was that Achilles had armor all over his body except in the back of the lower part of his leg, and that's where he got cut. And,
and that's where this stinger has to precisely inject.
Yeah, there you go.
Yeah.
Okay, so now we get to the fun part. Let's talk specifically about the jewel wasps. I'd like you to take us step by step through. It's a whole orchestrated dance between the wasp and the cockroach. And as I've referred to several times earlier in this episode already, it comes straight out of a horror movie. So why don't you tell us about the dance, beginning with that first sting?
Strange enough, when I was starting, I was always kind of surprised on how avid hunters the wasp is when you throw a cockroach in with it in their breeding cages, where they're not aggressive or territorial with me or whatever. Like if I go in there, they'll try to escape. But as soon as they sense a cockroach, you can see the wasp antenna going. It orients itself towards it and starts to try to flank around the side of the cockroach. And antennas start going. You can see it get real stimulated. Its posture raises, and then if it sees an avenue of attack, it will move quickly to grab with its mandibles the cockroach, and then the cockroach then starts like a rodeo and will start kicking and jumping and moving around, and in some cases will kick off the wasp. But if the wasp is quick enough, the first sting goes to the thorax, prothoracic ganglia, and that injection is GABA, a known inhibitory neurotransmitter. And that causes this kind of flaccid paralysis of the front legs. That should do it, you would think, right? But GABA is such a universal, well-controlled neurotransmitter that the cockroach's internal physiology will clear it within a minute. It'll take care of it. Yeah.
Okay.
After that, though, during that minute and the front legs can't really interfere anymore, that's Yeah, please.
Can I pull out a nerdy detail from you as well about that first sting? So you're talking about GABA, and just for some in our audience are science nerds. I'm certainly a science nerd, and I like to dig into these things. So this GABA, it's activating chloride channels, electrical channels inside the nervous system of the cockroach. We have chloride channels as well. We respond to GABA. And the other thing, you mentioned the GABA, but I understand there's a mixture of components in the venom. GABA is the one, taurine and beta-alanine amino acids are excitatory amino acids. And I read that there's an optimal ratio of these things in the venom, a ratio of 10 to 7 to 4 of these various components.
All three of those are what are called GABA receptor agonists. So GABA is the well-known GABA receptor. The receptor's named after it. But beta-alanine and taurine will also, they're slightly different in shape, but they have a positive charge on one side, negative charge on the other. They will activate these receptors. And optimal kind of depends on what your goal is. But if it's for a certain kind of longevity or intensity, you want it to last for about as long as the sting takes into the brain, just to keep the front legs from fighting back. But those are all GABA A receptor agonists.
And the reason I wanted to put that out is not just to show off, some factoids or whatever, but because this is a highly evolved dance again. And even at the first step, that first sting is not just some random collection of possible toxins. It's a careful ratioed mixture with a very specific ratio of these things. Very highly evolved venom.
Agreed. Yeah.
You were on the way down to sting number 2.
Yeah. So then now that it has an opportunity and it has a window of time, but the window is narrow, the cockroach will recover after that first sting within a minute or two. Then it takes the stinger, rather sophisticated ovipositor, finds a weak spot right underneath the head capsule and injects directly into the 2 ganglia that are in the brain, or in the head, excuse me. The biggest one, the one that we're most interested in, is called the brain. And the center of all this integration of sensory data goes into a place in the brain called the central complex, which is a complex right in the middle. So that's where the venom is targeted. And besides that, underneath it is a ganglia called the naphal ganglia. It used to be called the subesophageal ganglia, but naphal for being where the mandibles are. But that seemed to get a hotspot for venom too. And our experiments in both the naphal ganglia and the supraesophageal ganglia, the brain, both are important and both can contribute to locomotion. One is on the way down to the thoracic ganglia anyway. So to inhibit, you can control both. Both are important. Both are distinct ganglia, and both are targeted by the venom. And it takes about a minute. And when you're watching the videos, it's an awkward, slow minute where it's positioning this ovipositor and injecting these. There's this really nice paper that came out while I was still in grad school from Liebersad's group in Ben Gurion, who actually got my PI, my predecessor, into the project. He has most of the papers on this subject.
Ryan is about to describe an experiment that was done that involved agarose beads. Agarose beads are basically round globs of jello, which you can make in a certain way to have more or less density, more or less hardness. Because different organs inside a cockroach will have different densities or hardness, just like a piece of steak will be a little bit tougher than a piece of liver, and a piece of poached egg will be even less tough yet. The goal in this experiment will be to implant agarose beads of different density into a cockroach body in order to see if a jewel wasp can stab its ovipositor, its egg-laying stinger, into the cockroach and feel around to find where the ganglia is. Basically find the ganglia by feel, and then inject the venom into the bead with the same density as the ganglia. It might help to imagine yourself reaching around underneath the big heavy living room couch, trying to find the ball of yarn that rolled underneath it, and you come across a hairball that your cat just barfed up, and your kid's tennis ball, and you can tell the difference between those just by feel.
They have this paper, it was amazing, where they took the cockroach brain out and replaced it with agarose beads of various density. If these beads had a similar density to the cockroach, it would inject venom into the bead. If the bead was too hard or too soft, eventually the wasp would abandon the animal. So without saying too much about it, they found some mechanosensors on the stinger.
Mechanosensors are the sense organs that feel touch or pressure. You've got lots of mechanosensors in your fingertips.
And it looks like what it's doing during that time is finding by touch or by density, somehow identifying the right spot of the brain and injecting venom directly into it. Wow. And they also have this kind of elegant paper using radioactive wasps that were fed radioactive amino acids to make the venom out of. Then they go look in the brain to see where the radiation was. And it was concentrated in the central complex and in the subesophageal ganglion. So it's pretty clear that through these two papers that it knows where it's stinging and is precise in its sting.
Right.
And right into the brain, very little outside in the head capsule. So it's not just randomly injecting venom anywhere. It puts it right where it's most important.
Wow.
And one of the things that does, I understand, it shuts down the escape reflex, and there's a number of other behaviors. Maybe you want to tell us about that.
Yeah. So after the sting occurs, in our hands, at least in the lab, it took about 3 hours for it to be maximally effective, where the escape response basically goes down to zero. But during this time, it's not paralytic, which is strange. Even though GABA would be paralytic for about a minute, that wears off. There's some good papers that describe dopamine as part of the venom, but that's not part of the hypokinesia. It seems to inspire the cockroach to groom itself for about an hour or two. So after the sting, the cockroach kind of recovers and is a little, I don't know, dopey maybe, not to turn a phrase too much, but is using the dopamine that starts to groom and is more content to sit there. But most fascinating to me, it is clearly not paralyzed. It's grooming itself.
Like a cat licking its paws.
Yeah. And during that time, the wasp finds some suitable burrow nearby.
Right.
And then comes and checks on the cockroach, who will poke at it, it will grab it with its mandibles. And if the cockroach is still a little refractory or, has some adverse reaction to it, it gives it a little bit more time.
The turkey's not done yet.
Yeah, the venom's not fully kicked in. And when it comes in and finally grabs it, and it seems like it could start manipulating it, it will grab its antenna quite precisely, rip off its antenna, take a quick drink of leaky hemolymph out of the antenna.
Hemolymph is basically the insect's blood.
Still unclear about that. There's some speculation it's tasting venom. I don't see, I love the idea. I just don't see any evidence for that yet. As far as I'm concerned, it's taking a drink of the hemolymph.
Right.
Maybe just it's thirsty. Then it will grab the antenna nub and start dragging the cockroach. And this is probably the most important aspect to the reproductive cycle, is now that the cockroach is fully under the influence of the venom, yet not paralyzed, it is, I would characterize it probably a little too anthropomorphically as lethargic and apathetic, but not paralyzed. It doesn't care. It doesn't want to go, but it's happy to begrudgingly follow the tugging of its antenna to wherever it wants to go. And then wherever it had this previously identified burrow, the wasp will drag it slowly, but under its own power, just directed by the wasp. The cockroach walks to this burrow, and then that's when the wasp will lay an egg on it. Just one egg on behind the second leg there. Somewhere a little out of reach, because the cockroach will still groom itself and will go through sleep-wake cycles, more sleep appearing than wake, but it will go through cycles of activity inside this burrow. And we've had some videos we were taking. It seems like when we were watching this occur, it was about 50-50 chance that the cockroach would identify the larva growing on the outside, the egg developing, and then would just eat it. And then 50% chance that the larva develops all the way and finds its way inside, goes from ectoparasitoid to endoparasitoid, I suppose, and then eats the internal organs. That's when it is lethal to the cockroach.
And before we get to that part, so the adult wasp has dropped the egg under the armpit, as it were, of the leg, and then it leaves, but it doesn't just leave, it actually seals the door. You want to tell us about that?
That's right. Yeah, sorry, I jumped ahead too far. So it goes from neurosurgeon to mason. I don't know how else to put it, but then it will go find random debris, miscellaneous stuff, and it will just kind of barricade the door to keep the cockroach from predation, so anything else can go in there and eat it. It's not so tight that the cockroach couldn't get out if it wanted to. If the cockroach really tried to find a way out and had its own power and impetus to escape, it could power its way through. The adult wasp, in about 5 weeks after that, will find its way out. The cockroach could. It just kind of keeps it protected and keeps it safe. And then, the adult wasp leaves. And then the egg will take 2 or 3 days to hatch. Then it becomes just this little larva with piercing mandibles, and it sucks blood off of that armpit region, as you described. And then that gives it enough material to go to the second stage, which is a bigger, more noticeable. Those have kind of cutting mandibles where they can cut their way into the cockroach cuticle at that point. And
Which
then
is still alive.
Yeah. This is about 5-6 days later. Then once it goes inside the cockroach, it goes into its final form of the pupa. The third stage, the third instar, is the fancy name for it. That then is the stage that will pupate, and it needs a lot of material to do that. And so this third stage will eat all the internal organs in a kind of brutal selective order, where it will keep the gut basically alone, and will eat all the musculature and whatever it can get a hold of, the fat body and all the high calorie good stuff, and keep the gross gut alone, and part of the ventral nerve cord, it appeared. And then lay down silk inside and pupate inside. There's another paper that came out from a group from Germany, I think, that identified, using some organic chemistry, fungicides and antimicrobial compounds that this larva is secreting meanwhile to sterilize this area and preserve the cockroach husk. And then that eating the organs is, of course, fatal. So up until that point, the cockroach is lethargic and content, apparently, to sit there, because it could escape if it had the power to escape. It has the physical power, just not the mental capacity, I don't know what to say about a cockroach's motivations, but it doesn't. And until the moment it is too late is when the parasite goes inside at the third stage and eats the internal organs, sterilizes it, makes a pupa, generates a rather hard cocoon on the inside. And then the cockroach is not rotting because it's sterilized, which is nice. Because that could be deleterious to the wasp. But then it takes about 4 weeks if it's a male wasp, 5 weeks if it's a female wasp, to develop from the third stage larva to the full adult wasp in that pupal stage. So maybe 7 days, 7 days, I forgot, but about that order of magnitude to go from egg to pupa, and then 4 or 5 weeks from pupa to adult wasp. The cockroach is alive for the first part, is just a home as a corpse for the second part.
And can we linger on that for a second? Because I had, in the bit of reading that I did, I had thought that the reason the larva was very selective in what it eats was because it was trying to keep the cockroach alive as long as possible.
Yeah.
But now you're making it sound like maybe part of the reason is it doesn't like the taste of the intestines.
Yeah, because that becomes speculative again, and that's a hard experiment to ask. But I would suppose, just to put that caveat on it, that yeah, keeping the cockroach for alive as long as possible keeps it from rotting and keeps the food fresh. That
Right.
makes sense.
Yeah.
So that was our first idea, and that's probably a reasonable hypothesis. Then the second, once we saw that the gut was separated by a barrier of silk and basically untouched, and we thought, well, maybe it's doing both of what you said. The other one is staying away from what might be infectious, or I don't know, somehow or another ruining its microbiome or something that would prevent its growth in some way. Choosing the best parts to eat, keeping the grosser parts for later, maybe keeping the cockroach alive as long as possible. All that is probably reasonable.
Right.
Yeah.
And then it crawls out. It cuts a hole and bursts out like in the movie.
5 weeks later, it chews a way out of its cocoon, then out of the husk of the cockroach, then finds its way out of the burrow and can complete its life cycle. Within a day or two, maybe 3 days, it is competent to hunt. It doesn't need to be taught how to find cockroaches. It doesn't need to be taught how to precisely sting a cockroach. But we feed the adult wasps a very pleasant meal of water and honey. So really the cockroach is to feed the babies.
Okay.
Yeah. I was going to say, it's almost to me like it's like they're missing this piece of their life cycle, basically like pupation. Like a caterpillar doesn't need anything external to pupate, right? Is that right? Like they could basically go from a caterpillar to a pupa to
and they're getting their fuel essentially from the leaves that they eat. But in this case, it needs to get fuel. Wasps and bees will do this too. And when you see the honeycomb of bees and you have a little pupa in there, they're getting fed. But they still have to go through the stages, but they're getting the colony is taking care of them. They're being fed and taken care of and reared. In this case, it can't leave the husk of the cockroach, but it has its own food supply. It's surrounded by its own banquet, as it were, of meat.
Wow.
Yeah.
Yeah, that's amazing.
And so I want to talk about two things then. Again, I'm trying to understand the evolution of these things. So on the one hand, the venom. So we talked about the venom containing these neurotoxins, the GABA, the beta-alanine, etc. Is there other things in the venom? In the snakes, we talked about the many, many kinds of components in the snake venom. But in these wasp venoms, is it only neurotoxins or are there other things in there?
Oh, yeah. That's where my research is now, is trying to look at these other components. The small molecule neurotransmitters, they were, I suppose, easy to hypothesize what they do. The protein, the proteome, as you were, of the venom has something on the order of 260 reproducibly detected proteins. Some are quite small, novel, small peptides, which are common for venoms. Most venom small peptides would induce some sort of pain, become some sort of channel modifier, or even lytic, destroy the cell membranes, or cause inflammation. But what we found is that none of these peptides seem to do that. When we look at the brain after the sting, there's no real sign of necrosis or cytotoxicity. There's no apparent damage, in stark contrast to, say, a snake venom, even though there's a lot that they do have in common, including some of the enzymes that appear to break down membranes and release factors. One's called phospholipase. A2 is pretty common throughout venoms. High hyaluronidase seems to break down the extracellular matrix components, and that's common across venoms, across taxa, and snakes have this in common with ampulex. But they don't seem to destroy the brain. And that's where I kind of am interested now, is what about this venom is modulatory. It changes the behavior. It changes the way, the response to integration of signals, including hunger, thirst, light cycle, day cycle. Some speculation, I like this idea from Lieberstadt's group that they were narcoleptic almost, that they're mostly sleeping most of the time. And I'd like to see that assay. But what we ended up finding was a handful of additional neuropeptides and enzymes. And I think they make for good projects for students, because you can train them on the biochemistry and molecular biology and enzymology and all that stuff by selecting one of these enzymes, expressing it, cloning it, characterizing it, and seeing what it can actually do. In us, there would be inflammation, but it's hard to say there's a direct equivalent, but damage response pathways seem to be lit off by the venom. Maybe it will release antimicrobial peptides because insects have an innate immune system. They don't have an adaptive immune system. It appears some of these things are homologous with other pathways in Drosophila, fruit flies, that trigger an immune response. I don't know what's going to happen as an immune response inside the cockroach brain, but maybe it's doing something to prevent it from getting infected. It did just get stung in the middle of its brain. So we're going to try to look into that. We have an enzyme called adenosine deaminase, which is a growth factor in insects, and we have one too. We're seeing that might be neuroprotective because release of this compound ATP, the same ATP that you think energy currency of the cell, gets metabolized to adenosine. Adenosine appears to cause cell death if there's too much of it for too long. So this enzyme in the venom seems to keep it low. And if we keep the concentration of adenosine low, the brain will not die as much from the insult of the sting and the presence of the venom. There's a whole family of enzymes in there that are known in mammals to process neuropeptides. So maybe it's not the neuropeptides themselves, but the processing of the endogenous neuropeptides that seems to be important. Maybe it's breaking down its own. One is corazonin that seems to control reproductive behavior. One is tachykinin that controls locomotory behavior. So there's enzymes that control its own internal processes, and there's quite a few of them. So we have hundreds of these possible targets.
Wow. So that's already another one of the many things that I find so amazing in this story, just the evolution of this venom. Again, it's not just simply a wasp stumbling across one or two really cool toxins and using that. This is a very complex mixture that has evolved.
It seems so.
The other evolutionary thing that fascinates me, and this is a little bit more philosophical, and you may not be necessarily haven't been thinking about this in your research, but the evolution of the programming. I used to think it was amazing just how, like monarch butterflies, they have this pre-programmed migration route, and I thought, wow, isn't that amazing? But now this whole sequence of events that have to happen in the right order, in the right way, in this stinging, the two stings and the leading, you brought us through all of that. But even with the larva being pre-programmed to not eat certain internal organs, eat other ones for good reasons. So this pre-programming, how would that evolve? How is that found in the brain?
I do think about it a lot. I do. I just don't know how to ask that question at a bench. Yeah. So the way I think about it is that has to be part of the nervous system. That network of connections, the way we have our own innate behaviors, right? We can get up and walk around. Toddlers learn to walk at a certain set point, right? These developmental processes take place. But when we think about ourselves learning and thinking and changing who we are, and as our personality changes, that's plasticity, And so it has some fundament. And that has to be encoded genetically. That gets expressed in the nervous system, the nature part of nature and nurture. It's there. It can't not be there. How to start looking for that as a pattern and interrupting it and experimenting with it, I have no idea.
Right. Yeah.
But I think about it often. And it's amazing. It is amazing to me that it's there in the pattern of neurons.
We're hearing a lot these days, even at the lay level, of course, because of this explosion of in air quotes, artificial intelligence. I don't know if any of us knows what that really means yet, but anyhow. But, we're learning a lot about, and I actually did take a course years ago on neural networks. I do have a background in software development. And so we've learned, at a rudimentary level, at least, how these neurons work and how they fire. And so we train these systems. So, where is the training? What created that neural network that now comes kind of it's like a boot routine on a computer. A boot routine is built.
It's absolutely a boot routine when you want to think about it like that. That's a good point. Yeah.
So that's interesting. So is that an area of research at all, or?
Well, not that I'm aware of, but I would put it on a scale of geological time. And philosophically, I'm sure evolutionary biologists, my wife is one, she'll probably kind of scoff at this idea, but I love the idea that in neuroscience fields, they talk about learning and memory. We learn a new behavior, a new fact, and then we can recall it. DNA and evolution, learning and memory. You learn something by some mutation, sure. If it's preserved in the DNA, is that a form of memory? It's one that takes learning by random chance and experimentation, sure. It's not directed, fine. But when you multiply it by 250 million years, you can't imagine the magnitude of failures to see the one success. So, and then it's encoded in the DNA, and little by little. But where did the first haphazard leap come from where it kind of injected a brain and kind of, that leap, that quantum leap, I don't know if that took 5,000, 10,000 years. How could, the life cycle 1 year at a time, you get one shot at it every breeding season. Where did that quantum leap come from? Where now it's injecting cockroach brains from, where several tens of thousands of generations before it wasn't. I don't know. You know what I mean? The only reason why I can feel comfortable around it is the multiplication by the time span that it had to do it.
Right.
Otherwise, I'm baffled by the idea. I'm compelled by it. I don't know how to even look in the fossil record for such a thing. What would we look for? We just see the progression of anatomical features. We can describe them. We can speculate on them.
Right.
But that's amazing. It's a crazy question. I love the idea of it. How does it know? How does it know? I don't know.
Yeah.
There's another example, I guess I'll say, of this pre-programming and evolution, but now not in the wasp, but in its prey, in the cockroach. You were mentioning earlier how the wasp is carefully maneuvering around the cockroach, trying to get the right angle of attack. And the cockroach, in turn, is recognizing that, and it has a whole pre-programmed response to that happening. I don't know if you want to say a few things about that.
Yeah, so the next logical leap would be, okay, well, if there's this evolutionary arms race, what kind of counter maneuvers is evolving in the cockroach? And then my counter to that would be the idea of pest control. This is anecdotal. I don't remember seeing this, but as a scientific report or something, but Ampulex compressa was introduced to Hawaii to try to combat the cockroaches there. Perfect habitat for Ampulex. It thrives at home there, but it didn't put a dent in the cockroaches. So one of the necessary aspects of evolution is to eliminate the cockroaches that were not stung, or the ones that weren't adapted. And there's so many cockroaches, and their life cycle is so, robust, that you're not evolutionarily steering them, because so many survive without ever encountering a wasp, that the ones that may have found a way to escape don't contribute to the next generation as much as the rest of them do. So I bet you if all cockroaches were being under pressure from this wasp, there would be some trait that would evolve at some point. But the cockroach's success seems to be that they can live anywhere and eat anything and still be pretty fecund, if that's the right word. And the wasp just seems to take advantage of their success.
Yeah, it's kind of, I guess it's a numbers game for them. They can just out reproduce the effect of the wasp. That's fascinating, because that's what I was thinking of. I'm thinking to myself, okay, they've had, 200 million years?
Well, for the cockroach and this wasp in particular, let's just put it on the order of 100 million, maybe.
Okay, so they've had 100 million years to deal with this. Right. What's wrong with these cockroaches?
Innumerable generations.
Right. And they haven't, because they haven't needed to. They've survived without needing that. Okay, that's interesting.
Yeah, and even though we don't have a statistically significant set of this experiment, but a few students have tried, though it does take a while, because the cockroach is quite long-lived for insects. It takes a year to become an adult, and then they'll live for another year as an adult. So we allowed the cockroach pretty young, within a week of it becoming a mature adult cockroach, get stung. Then we made sure that there was no egg on it. We pulled whatever egg was off or interrupted the sting after we saw that the brain sting was clean. And then we kept them isolated to see if there was any kind of long-term damage, or did their behavior come back. We knew it would come back. They would recover in about a week or so. But what about long-term? And we didn't see any difference. It seemed to be that they lived long cockroach lives after being stung. And in fact, the small data set that we did have, and it would be nice if somebody would want to take this project up again, is that they said live a little longer. They didn't seem to be cut short at all. And if it was statistically significant, the longer, more healthy lifespan after the sting, I would love to have had that data to see. But at least we know that probably the sting itself isn't deleterious. So if it did escape, or it did eat the wasp larva and managed to escape after a few days, it could probably go off and live a normal cockroach life. And then maybe it had some mutation itself, where it was protected against the venom, it could get away faster, can detect the growing larva in some way. And that is a mechanism over time that can lead to protection against it. But if 9 out of 10, 99 out of 100 cockroaches aren't facing this issue, it probably is not a trait that will appear. So it's a really interesting idea. I love the idea.
Yeah, so there's basically there's not pressure, I guess.
Yeah.
For evolution, you need some sort of
Yeah.
pressure, right? Some sort of environmental pressure or something to drive it forward. It's not just going to happen.
Yeah, it's not enough just to have the mutation. You have to select for that mutation.
Right. Okay.
And that takes time.
Very good. That takes time. Yeah.
Well, Ryan, we wanted an expert's description of what's going on with this predator-prey relationship. You've given us a really great deep dive. Is there any part of the story, that we didn't cover? You might want to have a few last words on that whole story.
Just where my brain is now is my students and I am getting interested in this idea of neuroprotection. That's something that's a new kind of paradigm where venoms were neurotoxic. They're even described that way, right? That's in their definition. And we don't see that as this venom, where most venoms are going to kill the cells or at least inflict damage and cause inflammation and pain. Maybe there's an aspect to this one that while it's manipulating behavior, other aspects of it might be protecting the brain for as long as possible or trying to at least mitigate the damage associated with the sting. You don't want an infected brain and you don't want a dead cockroach before the larva develops. So that's where I'm at now. And I'm hoping in the next several years, that's where our work will go.
Excellent.
Yeah, that's really unique. I would think that type of toxin is pretty unique, I guess.
And who knows what we could discover? Something about it, that some novel peptide that keeps a cell from excitotoxicity, which is a problem in all neurons, even in mammals. Something about it that keeps it alive longer under stress would be great to find. The hope is to find a leap to humans, right?
Yeah.
And the biology of it, I would be content to just understand the biology. I would be ecstatic to find a connection to mammals.
Yeah, that was my next question. How does this research possibly feed into on a human level, and dealing with, the more we learn about, I guess, the brain and the things that affect the brain and the toxins. I think of things like Alzheimer's and so on. These are, I guess, I think there at least is a theory that Alzheimer's is a toxin.
The beta amyloid, sure.
Yeah, the beta amyloid. Yeah, right. I guess I'm a fairly big picture person just in general. And so I love that generalization, how everything's connected from the venom of a wasp to the working of the human mind. There's connection there. It's not just, that's fascinating.
That is my hope. And there's precedent for it. There's a lot of the pathways that have been discovered in mammals as critical were first discovered in insects. So there's homology there. There may be, I'm sure there's things that we have yet to understand that if we find them in a more simple model, I'm careful to say simple because once you look at the cockroach brain, it doesn't, but to compare it to a human brain, it's simple. There's things going on there that maybe reverse engineering this wasp venom will identify new pathways or new approaches or new ideas. But even not, there's spider toxins that can treat chronic pain by being calcium channel blockers. Somebody was telling me about a scorpion toxin that has a unique proclivity to bind to human tumors, maybe not to kill them, but at least identify them for surgeons to excise. So whatever we find, maybe it won't be the direct connection to adenosine or tachykinin pathway in the human brain, but it might open a new paradigm, and that's what's compelling to me.
Right.
So, Dr. Arvidsson, you've written quite a few scientific papers. Are there any books in the pipeline, or maybe is Ridley Scott reaching out to you for the next movie?
Nope. Just when we get enough data, my students seem to, which is nice. I've made the transition. Most of my time goes to teaching and mentoring now. But when my students put together enough, I get real excited and I get my neurons firing, and I try to wrap it up and try to get it out. So there's 1 or 2 papers coming. We have new projects that have kind of got away from this venom. We're looking at the metabolism of the cockroach brain. It seems to have its own unique way of handling food and energy demands. So, just probably will never get away from this Ampulex toxin. Just one more peptide, one more new thing in it that hasn't been explored yet. Can we see if it's doing something? Can we just see what and then apply it to a simple human cell line or something like that? Yeah, just
Well,
I'll probably work on it until I'm retired.
I can feel the excitement in your voice there.
Yeah.
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 It's been great talking to you. I really appreciate you
thank you.
giving us your time at this. It's a very busy
Yeah.
time of year for academics, I know, and I thank you for giving us your
Yeah.
time.
Of course. Thanks for the invite. It means a lot to me because it's something I'm particularly excited and interested in. And very happy to take time out to think and talk about it. And thank you for your compelling
Yeah.
questions. I'll probably be thinking about it for days now. Trying to think,
Good.
Yeah.
That got me thinking.
Excellent.
That got me going.
Thanks, Ryan.
Yeah. Thank you.
All right. Have a good day.
Yeah. Yeah.
You've been listening to Recovering Evangelicals with your hosts, Luke Jansen and Scott Dyer. Send your comments and suggestions for new episodes to lukejjansen@gmail.com. Join our private discussion group on Facebook by searching for the Recovering Evangelicals podcast group. Look for the logo that you always see at your favorite podcast catcher and ask to be let in. Subscribe to lukejjansen.wordpress.com if you want to get advanced releases and group posts, or browse through our archive for a full listing of all our previous episodes. And please help the podcast by sharing with a friend and leaving a review at your favorite podcast catcher.